Hey, good morning, everybody, and welcome to the 2022 virtual edition of the Royal Tyrrell Museum Speaker Series. I'm Caleb Brown. I'm one of the researchers here at the Royal Tyrrell Museum. Today, the Royal Tyrrell Museum and its cooperating society are very proud to present Jade Simon. Sorry, one of my colleagues is Jade Scott. Jade is a PhD candidate in the Department of Ecology and Evolutionary Biology at the University of Toronto. Jade is originally from West Virginia in the US, where her interest in paleontology was sparked by road cuts and crinoid fossils. She completed her BSc in geology and a BA in anthropology at West Virginia University. Soon, her passion for paleontology drove her to Montana, where she completed a master's degree in earth science at Montana State University. For her master's thesis, Jade studied giant theropod dinosaur eggs from southeastern Idaho, with implications for taxonomy, paleobiogeography, and reproduction. Following her master's, Jade moved to Toronto to start a PhD at the University of Toronto, based at the Royal Ontario Museum. Her doctoral thesis investigates the ecology, growth, and diversity of theropods along the bird-dinosaur transition, or the dinosaur-bird transition. This specifically involves the study of the microscopic structure of fossil bones to reconstruct growth and species diversity of Oviraptorosaurs, which is a group of feathered bird-like dinosaurs. And it's some of this research that Jade will be sharing with us today. Throughout her research career, Jade has done fieldwork in Alberta, British Columbia, Montana, Idaho, and China. In addition to her research, Jade is also passionate about science outreach and disability advocacy. Along with her service dog, Basil, she has presented in classrooms, distance learning programs, and professional society conferences to advocate for increased accessibility for disabled students and researchers in academia. So without further delay, I'll pass control over to Jade Simon.
All right, thanks so much. I'm going to set up my screen share really quick. All right, thank you so much for inviting me. I'm very excited to be presenting my research for you all, and I will try to highlight some really cool specimens from the Royal Tyrrell that I was fortunate enough to include in my study. So as Caleb mentioned, I work on theropod dinosaurs, and the group that I really focus on right now are Oviraptorosaurs. So I'll start with just kind of an overview of what are these very silly looking dinosaurs, how do they fit into the context of other theropods, and what sorts of questions can we ask about them. So Oviraptorosaurs are a group of feathered bird-like dinosaurs. They vary pretty greatly in size. We have some very, very small. We also have some that are nearly the size of a T-Rex. And they are known from all across Asia and North America throughout the Cretaceous, but mostly towards the mid and late Cretaceous. They're a really interesting group of theropod dinosaurs. You can see in these reconstructions, life reconstructions, they are very silly looking. They're charismatic to me because of that. So some of them have crests on their heads, but you'll notice in these images, they don't really have teeth. So not very many of them, only the very earliest ones. So most of them have a beak, no teeth. Some of them have these crests. We do think that they were all feathered, and they're just a really interesting group of dinosaurs. So we can see here looking at this phylogeny, they are pretty closely related to the theropod dinosaurs, where we get the transition into birds. So they're in the Maniraptoran group, so closely related to troodontids and dromaeosaurs, and along that dinosaur bird transition. And there are two main groups of Oviraptorosaurs. And I'll just talk about this a little bit because I focus more on one group than the other. So I want to get you up to speed on the lingo. So the Oviraptorosaurs are split into two main clades. It's the Oviraptorids and the Caenagnathids. Caenagnathids are the group I primarily study. They are mostly found in North America, and they are a bit of a question mark. There's lots of things we can ask about Caenagnathids. Oviraptorids are found mostly in Asia, And we know a lot more about them because there are more complete fossils known for oviraptorids. So I'll get into some of the differences here. Caenignathids tend to have a more shallow jaw and much longer hands and feet. So the bones of the hands and feet are much more elongate. And oviraptorids have an arched, more compact, shortened and high jaw, and then shorter digits in the hands and feet. And those differences, mostly in the skull, the jaw, and in the hands and feet, those are probably reflective of environmental differences. So there's some differences in the ecology between the caenignathids and the oviraptorids. And we'll talk a little bit more about that in just a second. Oviraptorids are very well represented from several complete specimens found from across Asia. So we have lots of complete skeletons, beautifully articulated, gorgeous specimens. And that's why we know a lot more about their anatomy and also just about their general biology. Oviraptorids are a bit famous for their association with eggs. You've probably seen some images of dinosaurs on their nests, and often we're looking at oviraptors in those reconstructions. So the name oviraptor means egg thief. They get a little bit of a bad rap, but that's because the first oviraptor was found, it was a skull found associated with an egg. We later figured out that that egg was not prey, but it was actually the animal's own egg. So they were known to sit on their nests and care for their nests, but we're not sure if they cared for their young once they hatched. That's a bigger question. But there are lots of specimens of oviraptorids sitting on their nests, and those are pretty incredible fossils. And that's what I studied for my master's, were some eggs that were from North America. They weren't associated with a skeleton, but they were from a giant Oviraptorosaur, which was pretty neat. The caenignathids in North America, we have a bit of a different picture. It's much more fragmentary. We don't typically have these nice articulated specimens like you see for the oviraptorids. So caenignathids are a little bit harder to figure out. We have bits and pieces from all across North America. We have sometimes a jaw, sometimes pieces of the hands or foot. Very rarely, though, do we actually have specimens that have different parts of the body preserved together, unfortunately. So that's one of the reasons that caenignathids are such an interesting puzzle. So there's lots of questions that we can ask about Oviraptorosaurs in general, but especially caenignathids, since their fossil record is a bit more fragmentary. And this is why I get so excited about them, because there's lots of stuff to try to figure out. So caenignathids can be a bit of a big old question mark. We have lots of things we can look into, like what kind of reproductive habits did they have? What kind of environments were they nesting in? Did they care for their nests the same way that oviraptorids did? Did they care for their young? What kinds of environments were they living in? Are they more adapted for wetter environments, closer to river systems compared to oviraptorids, which are found in much drier ecosystems? There's even a lot of debate over the relationships of the different caenignathid species to each other. Because they're so fragmentary, it can be hard to figure out which species are more closely related and what are the family groupings like there. And the diversity, what does the diversity of the group look like? So how many species, and as we look through time, were they diversifying and then slowing down in their diversity, or what was really going on? There's even questions about the diet of Oviraptorosaurs is pretty debated. So it's hard to get a very good straight answer on whether they were carnivorous or omnivorous or herbivorous. There are arguments for all sorts of things like mollusk eating or strict herbivory or egg eating. That one is a little more settled. They're probably omnivorous, maybe herbivorous, but lots of different questions to dive into, which makes them a really fun group of animals to study. Now, fortunately, there have been lots of new discoveries of caenignathids in North America in the last 5 to 10 years, and that has included some more complete, though not entirely complete, specimens. So one of my favorites is at the Royal Tyrrell Museum. This is Apatoraptor pennatus, and it was discovered, or well, it was described in Funston and Curry paper in 2016. So that's Greg Funston and Phil Curry. And this is a wonderful specimen. I was very, very lucky to get to see it myself on a visit to the Royal Tyrrell Museum a few years ago. And it's really exciting. This is one of the only specimens that we have different parts of the body actually in articulation for a caenignathed in North America. There is another specimen where we have different parts of the body in association, and that is Anzu wiley, which is a pretty remarkable specimen as well. And that's housed in the Carnegie Museum. This is a kind of medium to large, kind of person-sized caenignathed, and you may have heard about it if you are a dinosaur fan. It was pretty big news in 2014 when this came out, because this was the first caenignathed that wasn't just bits and pieces here and there. It was actually a good amount of the skeleton. And then we got, thankfully, Apatoraptor. And we do also have at the Royal Tyrrell Museum, you will have Chiristinotes progressilis, which has jaws and hands as well, but they're not all from the same articulated individual. We have kind of bits and pieces from different animals. So there also have been lots of fragmentary new finds. So we have partial skeletons that have been found. We have hind limbs, so legs and feet. And these are all new discoveries that haven't been published on. So these are all from across North America in the last 5 or so years. So it's very, very exciting, lots of new specimens, and that opens the door for a lot of different avenues of research on North American caenignathids. So right now, this is the kind of rough overview of what the distribution of caenignathids looks like. So the size just indicates the relative body size. The really little ones are small caenignathids. The really large one is the giant caenignathid that we have an occurrence based on eggs from Idaho. And so you can see we have lots of different body sizes, and we actually have a number of specimens. So these silhouettes represent individual specimens, and this is ranging across the latest early Cretaceous all the way to the end Cretaceous. So the kind of Aptian Albian all the way to the end at the Maastrichtian there. All right, so for my work, I basically go around and look at as many of these specimens as I can get a hold of, and I study the bone under the microscope. So I do what is called palaeohistology, and that is the study of fossil tissues under the microscope. It can also be called osteohistology, so that's bone tissue under the microscope. And I do focus on the later Cretaceous. So we will look a little bit at what the picture of diversity for Oviraptorosaurs caenignathids looks like at that late Cretaceous point in just a moment. But I want to talk a little bit about histology, how histology is done, just to kind of bring you up to speed on what that looks like, because it's a really fun process. I really enjoy it. So basically we have long bones. So those would be like a femur or a tibia in the leg. I focus mostly on those. And you can take that bone, you can actually cut through kind of the middle of the shaft of the bone, and you can make a slice and you mount that onto a slide. And then we grind that slide down until light can pass through the specimen that's on the slide. And then we look at that under the microscope. So that's sort of the actual process of how we get our data. And when we're looking under the microscope, this is an example of what we might see. So this is a section of a bone. So I've zoomed in. So we're just seeing the outer edge here and a portion of the inner section of the bone. And we look at a couple of different things. So we can look at all of those different lines and dots that you're seeing here. Those are openings for vascular canals. So bones are a living tissue. So they are a mineral matrix that makes up the hard part of the bone. But then there's all these blood vessels going through that bone, and that's bringing nutrients to the bone because the bone is a living tissue. It's cells that are actually growing and laying down that different mineral and remodeling, fixing little fractures and things like that. So those openings are all spaces where blood vessels used to be. So this is a fossilized bone, and we're actually able to see the structures that we would see in living bone if we looked at it under the microscope. So we look at the vascularity, those spaces, that can tell us a little bit about how fast the animal is growing. So here you're seeing lots of those lines and dots, and you can see they're kind of lined up all parallel with each other. This is pretty classic of very fast growing bone. So this dinosaur, this is a caenagnathed, is growing pretty darn quickly, we can tell. And then we look also at changes in that vascularity. So as we go towards the outer edge here, is the pattern of those like lines and dots, that vascularity, is it changing is a big question that we ask. And that helps us determine if the animal's growth was slowing down. In this slide, we don't see that. It's not slowing down. You can see there's lots of blood vessels here, or spaces for the blood vessels, all the way to the outer margin. So that's one big clue that this animal is growing really, really quickly, and it's not really slowing down. Another thing that we look for are called lines of arrested growth, or growth marks, either term. And those are, you can see these kind of, the first one here is a bit faint, but there's a line here, and there's a line just here indicated by those arrows. And those are points where the animal did slow down seasonally. So there's some kind of slowing in growth, and then it restarts again, and it leaves that mark. So we look for growth marks to help us determine the absolute age. So how many years old was that animal when it died? So this animal, we only have 2 growth marks preserved. So it was somewhere around 2 years old. It might have been 2 or 3. We can have growth marks that don't show up anymore. The very earliest ones, they can get kind of overprinted as the bone grows. But this is somewhere around 2 years old, and it's growing quickly and it's not slowing down. So we kind of combine that information, and here I've just highlighted the actual lag, and we would trace those lags all the way around the bone, and you can actually determine how much area, how much bone was created from one year to the next. We also look at that as a metric of, from one year to the next, was the animal growing more slowly than the year before? So you can look at the total amount of bone put down in a given year and try to decide that. So this animal is growing quickly, it's pretty young, so we would say that this is a juvenile. Okay. Now, there are other ways that have been used to try to determine how old a dinosaur was when it died, whether it was a juvenile or subadult or a skeletally mature adult. And those are things like what the bone looks like on the outside, the external texture, and sometimes how the different bones are fused together. But when we look at living relatives of dinosaurs, like crocodilians, they don't have very predictable patterns in those sorts of features. So this sort of osteohistology approach is really becoming popular, very common in paleontology more recently, as the most definitive way to figure out if you're looking at a young animal or a mature animal. And that's a really important question, because one of the things we do in paleontology is name new species and try to understand species diversity of different groups of dinosaurs. So when we're naming a new species, a really important first question we need to ask ourselves is the animal that we're looking at a juvenile or an adult? Because we don't want to base our new species definition on an individual that's actually maybe a teenager, right? Because the body shape and different features that we might use to identify the species might still be changing. So we want to try to base our new species names off of adult specimens whenever possible, which means histology is a really important tool in getting to that eventual interpretation of species diversity. And this is becoming a really popular method in paleontology, but it didn't used to be. It's fairly new because it is somewhat destructive. We do have to actually cut the bone, but the amount of information that we learn from cutting up the bone is so great that it's worth cutting up the bone. And we do make casts, molds and casts of those bones before we do any sort of destructive sampling. So all in all, it's very much worth the practice, and it tells us a lot more than we could learn by just looking at the outside of the bone. So some cool examples of this sort of histology informed diversity work are in Triceratops. So if you've ever been to or seen images from the Museum of the Rockies, they have a phenomenal series of Triceratops skulls going all the way from very small baby all the way to adult. So you can really see how the skull and the rest of the body might change as that animal grows up. And those changes through the growth of an individual animal are called ontogeny. So we want to make sure we understand the ontogeny of the animal and its ontogenetic status, which would be, juvenile, hatchling, juvenile, subadult, or adult, so that we can really get a better handle on true species diversity. We've also seen this work done with tyrannosaurs. So if you're familiar with Nanotyrannus, that has been determined to probably be a juvenile Tyrannosaurus rex instead of being its own individual unique species. Similar work has also been done with Pachycephalosaurs. So there's been the idea proposed that Dracorex and Sticky Moloch are actually kind of juvenile forms of Pachycephalosaurus. So there's been a few different studies established doing this ontogeny and diversity work in the last decade, and they're all pretty fascinating. So that is essentially what I'm trying to do for oviraptors, or more properly, caenignathids in North America. So just to give you an idea of my work, oops, sorry, my slides stopped advancing. There we go. I'm going to look a little more closely at a few formations in the very latest Cretaceous. So if we kind of focus in on, this is going to be the Montana and Alberta area, we can look at the Hell Creek Formation, the Horseshoe Canyon Formation, and the Dinosaur Park. And the animals I'm showing you here in this diagram, these are the established species of caenignathid in the latest Cretaceous in Western North America. So we have one named species in the entire Hell Creek, that's it, just on Zoo Wiley. In the Horseshoe Canyon, there's two named species. That's Apatoraptor, which I showed an image of a minute ago, and it's at the Royal Tyrrell. And then we also have Epicuristenodes, which is housed at the Royal Ontario Museum in Toronto. And then in the Dinosaur Park, we have three distinct species, Chiristenodes progressilis, Caenignathus collinsi, and Cytopes elegans. So if we're looking at this as our known species diversity, you might think, oh, it looks like caenignathid's became less diverse towards the end of the Cretaceous. For some reason, they're more diverse in the Dinosaur Park and much less diverse once we get to the Hell Creek. And all of these are specimens are presumed to be adult specimens. So these are supposed to be reflective of different body sizes for those species. So you can see in the Dinosaur Park, we have three different body sizes that are established as three different species. In the Horseshoe Canyon, you can see there are two different body sizes established as different species. And then in the Hell Creek, we just have the one larger caenignathid. So my question that I've been tackling for my dissertation is, are all of these unique species, are they all skeletally mature? We probably need to go back and check, because when they were named, there wasn't histology done on all of these. They were named at various points in time, often before histology was kind of a common practice. So that's one first big question for me is, are they all actually skeletally mature adults? And the alternative then would be, if they're not all skeletally mature adults, are the smaller body sizes actually younger individuals of larger species, right? So that would be sort of the alternative. Are we looking at very small animals are actually hatchlings, and the medium size are maybe juveniles, and the largest one are adults? Or do we have a mixture? Are some of the small ones actually adults and unique species And then maybe some of the other small ones are hatchlings of other large species. So it can get a little bit complicated, but that's the general question I want to ask. Are we looking at all skeletally mature adults, or are we looking at different growth stages? And then we can kind of tease apart, what does that tell us then about the diversity? Can we get a clearer picture or a more accurate picture of how the species diversity of oviraptors changes through the Cretaceous once we understand the growth stage data? Okay, so I've sort of gone back through and we've grayed everybody out. So we're going to start fresh with no more assumptions about ontogenic status. And I'm going to take you through some histology slides of various canignathids from these formations. We'll talk a little bit about the histology and how I interpret the histology to establish whether we're looking at hatchling, subadult, juvenile, or adult. And then we'll revisit this image and kind of fill in the gaps of what do we know is for sure a skeletally mature and probably a distinct species, and which ones of these are actually different growth stages, which brings up some questions about whether they're their own species or not. Okay, so we're going to start with, this is an image that I actually used when I first showed you the histology. I was just very, very zoomed in on it. So this is a medium body-sized canignathid. By medium, I mean not person-sized, maybe half the size of a person. Small is a little closer to like small dog sized, and large for our purposes today, we'll think of maybe onzu, a person sized. So we'll break it into those kind of three rough categories. And then you can see in all of the slides where I'm showing histology, just to help you have a kind of a reference, in the lower left, you'll see the basic size class, whether it's small, medium, or large, and then a little key for the color once we finish our interpretation of whether it's a hatch year, juvenile, subadult, or adult. And then over in the right, I'll also include what part of the body the slide actually comes from, because some of them are from a femur, some are from a tibia. I think I even have some metatarsal fragments in here. All right, so we already went through the interpretation on this one a little bit when I first introduced histology, but we'll talk about it again. Here we're seeing pretty fast growth. We only have 2 growth marks, and the vascularity does not really change. It's pretty dense, and the animal's growing pretty quickly. So this one we would say is a juvenile. It's not a hatch year. It's got a couple of years of growth, but it's not showing any signs of really slowing down its growth and getting into that more mature stage. Here we have the other kind of opposite end of the spectrum to give you an idea of what we're looking at in when we find a mature animal. So here we're looking at the tibia of another medium-sized animal, very similar in size to the one we were just looking at a slide from. But you see there are many, many more growth marks indicated by those arrows. And they start to stack up on top of each other pretty tightly towards the outer edge. Something else that we see that I couldn't show you in the other slide is the vascularity changes quite a bit. So here you can see that we have lots of canals, pretty dense vasculature, and there's lots of parallel tracks next to each other, and they're even kind of blending into each other, just lots of blood vessels here in the lower section. But as we look towards the outer edge, you see that reduces pretty quickly until we get into the last section here where there's very few canals, and they actually are going from the inside of the bone towards the outside. So the direction is changed. And now there are these radial lines going towards the outer surface of the bone. This is pretty classic mature theropod dinosaur bone. So bone histology can look fairly different from one type of animal to the next, but within theropods, this is very classic, like mature bone texture. So we would call this a skeletally mature adult. And one feature that we look for to determine whether an animal is skeletally mature is called the EFS. It's an external fundamental system. And that's essentially this little complex here at the outer edge where there are so many growth marks stacked up on each other that it really starts to look like plywood, and it's hard to even count how many there are. That's that external fundamental system. And we want to see that to be able to call something a fully skeletally mature specimen. So this is a very good example of that. Okay, here's a large caenignathed. So this is bigger than the ones we've been looking at. This is more close to person-sized. We do see several growth marks, but the vascularity is not really changing very much, and we don't have any hint of an external fundamental system yet. We have a very slight change in vascularity, and again, no external fundamental system. So this one would be in the subadult category. So it's not growing as quickly as the first animal that we looked at. The growth marks are getting closer and closer to each other. It's slowing down a bit. The vascularity is changing a little, not a ton. And so it's slowing, but not fully mature. So this is a good example of kind of that in-between state. Okay, so that was an overview of kind of how we interpret some of the histology and some of the specimens that I've looked at of caenignathids. Now I want to look a little more closely at some that are housed at the Royal Tyrrell. So I was very, very fortunate to get to examine and sample 2 different specimens from the Royal Tyrrell Museum. One was Chirostenotes progressilis, and that is known from a couple of different specimens. One is at the Royal Tyrrell Museum, and then another is over at the university. And I looked at the one from the Tyrrell because it has a tibia. Long bones are great for histology. So when we looked at that, we found something pretty interesting. This is an overview of the entire bone. It's pretty crushed because theropods have hollow bones and they get pretty crushed when they're preserved. But you can see there's 2 growth marks. And when we zoom in, there we go, you can see that the vascularity is not changing very much. This is an animal that's growing very quickly and not showing much sign of slowing down. And it has absolutely no indication of any kind of external fundamental system. So this is a juvenile. And what's interesting about that is Chirostenotes was established as an adult. It was thought to be an adult specimen, both the Tyrrell specimen and the university specimen, based on the external bone texture. And that was really important for naming it and talking about diversity in the dinosaur park formation, since there's the 3 different body sizes of caenignathids known. But when we cut this one, we actually see it's fairly young and it's probably going to get quite a bit bigger. But another specimen of Chirostenotes that has been cut by Greg Funston is mature. So that raises some really interesting questions of maybe one of these specimens is not actually Chirostenotes. Maybe it's something new. Maybe it's a juvenile animal of a different species altogether. Which is very exciting. So that is one of my current projects that I presented on at the Society of Vertebrate Paleontology conference last year, and I'm very excited to get finished up. And another exciting Tyrrell specimen is Apatoraptor from the Horseshoe Canyon. So Apatoraptor is one of the few articulated partial specimens of a caenignathid from North America, described in 2016 by Greg Funston. And I was very, very lucky to be able to sample it when, I looked at it while I was there, and then it was sampled for me, and I got to make a thin section. And when we look at this one, it's pretty interesting. So we only see 2 growth marks, but I don't have a full cross-section of the bone. I'm just dealing with a fragment. So we have to be a little cautious when we only have a fragment. And we see some more information here than just the growth marks. We also see a lot of remodeling. So that's where the bone cells have been kind of overprinted on by newer generations of bone cells. So they're remodeling the bone. And you see that along this margin on the inside here. And we do actually see a little bit of a reduction in the vascularity towards the outer margin. It's starting to change a little bit. So this is not a mature adult. This is actually somewhere between the juvenile and the subadult categories. So we only have 2 legs. which we would think might make it pretty young, but we do have a lot of remodeling. So that may indicate that it's a little further along in its maturity. And we have a little bit of a shift in the vascularity. But this is probably going to get a little bit bigger, this animal. It's not done growing. It's not slowing down as much as we might expect. And so that raises some more questions too, like, is it possible that Apatoraptor is actually a subadult or kind of a later stage juvenile of something else? Maybe the other species that's known for Horseshoe Canyon, which was Epicurostenotes, and that animal is a little bit bigger. So interesting questions that we can address with these Oviraptorosaurs specimens. And I just wanted to give an overview here. So the main point that I like to kind of make with my research is we can look at all these different animals. And for example, here we have every animal on this slide is a medium size class. So we're talking like somewhere between like half the height of a person, not very, very big, not as big as Anzu. So this medium size class, if we looked at all of them externally and their bone texture looks the same, they have very similar anatomy, you may say that they're all adults of the same species or all adults of different species, depending on which features you want to use to diagnose your species. But the more important question is are they all actually adults? Just because they're the same size class doesn't necessarily mean that they are. So when we look at the histology, we actually see all these medium-sized animals are actually all over the map on their ontogenetic status. So we're actually looking at a couple of juveniles, one subadult, and one very mature adult. So some of these animals are going to get much, much bigger in their adult stage. Some of them are going to get a little bit bigger, and some of them have reached their full growth. So it kind of illustrates the importance of looking at the histology before we try to dive into the question of taxonomic diversity and naming all of these different animals. Okay, so with all of this in mind, where do we stand with our question of species diversity? Well, when we add in some of the new species from the Hell Creek, the picture there becomes a lot more complicated. But when we look at the Horseshoe Canyon, for example, with Apatoraptor, we could be looking at a juvenile of Epicurostenotes. So it's possible there's really only one species that we know of in the Horseshoe Canyon right now, based on some of the data that we have in the dinosaur park. There's big question marks of what do we do with this Curostenotes data? Is there one specimen that's actually representative of a juvenile that's going to get bigger and become the size of what we call Candignathus collinsi? So does that increase or decrease our idea of species diversity there? And in the Hell Creek, we have lots of new specimens that we need to look into. And I didn't show all the slides for that because that's part of my dissertation I'm currently still finishing up. But once we look at all of those, how does that change our interpretation, right? So our species diversity idea through the end Cretaceous here might change quite a bit depending on the histology interpretation, right? So we could have, for example, in the Hell Creek, maybe we have a lot of different species of different body sizes, where some of them are representative of maybe subadults of the largest species, and then maybe some of the others are actually small-bodied adults themselves of unique new species that haven't been named yet. So lots of different possible interpretations there. And I won't tell you what I think the answer is because I'm still writing that up and it'll be my dissertation here pretty soon. But that essentially is what I do for my research, to try to untangle histology and ontogenetic status of different Oviraptorosaurs, and then how does that change our picture of their actual species diversity and how that species diversity is changing through the end of the Cretaceous. I want to make sure I thank everyone at the Royal Tyrrell so much, because the specimens there and everyone's help with getting histology samples from those specimens was really, really crucial to my research and raises some of the most exciting questions, I think, in regards to the Horseshoe Canyon and the dinosaur park. But I want to thank all of you too for being here and listening to my talk. And I do want to give a special shout out to my service dog. who is shown here on the slide. This is Basil. She is my service dog, and she goes with me everywhere, including in the histology lab, and she supervises me while I cut up dinosaurs for my research. And I think she has a good time. All right, so with that, I would like to, I guess, turn it back to the moderators and see if there are any questions for me about this research or about Basil also. I'm happy to answer questions about her.
Great. Thank you very much for that really cool talk. Nice to see the research that you've been doing over the last couple of years. So for those in the audience who have questions, feel free to enter them in the Q&A box at the bottom of the screen. We'll start looking at those in a moment. Before we do, I'm just going to do a quick shout out for next week's talk. Next week we have Dr. Hanneke Mayor from the University of Bergen in Norway. And the title of the talk is Birds Above the Hobbit's Head, Tales from a Small Island Far, Far Away. So make sure you tune in to see that exciting research as well. Just looking at the questions here. The first question we have is about those growth lines, the legs. The question is about whether that cessation of growth is caused by a certain phenological or a certain biological reason. Do we know what's causing that growth cessation?
Oh, yeah, so this is not specific to oviraptors or dinosaurs at all. Most animals, if you look at the bone, you'll see growth marks of some kind, and people just use different names for them sometimes, but those legs are usually annual markers of growth, and they do tend to correlate with just periods where the animal's growth has slowed down, whether that's due to seasonal changes or changes in food availability. There's all sorts of reasons that might actually drive that change, but it's really consistent across reptiles, mammals, all kinds of animals. Yeah.
Okay. The next question is about chronospecies. So given that you have the different formations, is there possible that you could have anagenesis or chronospecies also complicating this taxonomy?
Absolutely, yeah. And that's an excellent question. That's something that comes up with the Triceratops research as well, is when do you kind of call it a new species, right? If you're thinking about anagenesis and this gradual change over time, but in the fossil record, we only get snapshots and we have to do the best that we can with those snapshots. So that's a little bit more of a complicated question, but yes, there definitely could be some anagenesis going on across these formations, complicating those relationships. And I think that knowing the ontogeny and understanding the actual ontogenic series of some of these animals, where we have the few that we have multiple specimens for, could really help us answer that. So if we could get a better handle on what the growth trajectory of any of those canagnathids looks like from hatchling to adult, that would really help us understand what the changes we might expect would be. And I think that would help figure out a better, help us get a better handle on which characters on the skeleton should we use. So that is going to be important for understanding anagenesis and when we decide we have a new species. When we're naming a species, we use skeletal characteristics. But if we're not sure which parts of the skeleton change throughout an animal's growth, that becomes really complicated because it can be hard to determine which skeletal characters are important for naming the species versus which ones are really plastic and are going to change. So yes, I think anagenesis is playing a role. I don't think we'll be able to address it until we have a better handle on how the bodies of these animals are changing and which characters are flexible and which are not, which hopefully we'll be able to establish with some of my research.
Great. So just to clarify for someone in the audience who might not know, anagenesis is evolution that's non-branching within a lineage. So species A evolving to B to C, as opposed to cladogenesis, where you have splitting evolution. And this is a big topic right now in a lot of dinosaur paleontology. The next question is about the allometric ontogeny hypothesis regarding the taxonomy. Is there other data such as ichnofossils or coprolites that could help with that question?
Well, for oviraptors, I'm not sure. In general, it is very difficult to tie ichnofossils directly to specific species, because they really are just a record of the size of the animal and a general idea of which kind of animal. For instance, if we have a trackway that has 3 toes, we are going to probably say that's some kind of theropod. But which one is going to be a big question. It's going to be based on body size, which we are learning is not a great indicator of species. It's really just an indicator of size. So trackways and things, I'm not sure, because they are harder to pin to actual fossils. But maybe, I mean, if we can't, if we have very small footprints that we couldn't tie to anything else, that could indicate a younger animal, but it could also just indicate a small species. So I'm not sure.
We don't always necessarily have the answers. That's part of the point, I guess. The next question is about the actual making of the slides. And how invasive is it to make those slides? And is it possible to glue the specimen back together again? Kind of what does it look like before and after, I guess?
Oh, excellent question. I meant to include a picture of that and I completely forgot. It is invasive because we are taking a chunk out, but it's worth it. And you can make a mold and cast of the entire piece that was removed. And you can put that back together with the fossil. So when I've done thin sections, we do a couple of different things to make sure the record of the intact fossil is there and that we're losing as little information as possible and making it really, really worthwhile. So we'll do a mold and cast of the entire bone before any cuts are made. So the entire thing is preserved as it was before we touched it. And then we also, of course, take lots and lots of figures. I often will make a 3D model as well using photogrammetry. And then we'll take a chunk out of the middle. We use very accurate precision saws, and we actually use them at pretty low speed. So the amount of bone that is lost to the blade itself is minimized as much as possible. And then the chunk that's taken out, we'll make a mold and cast of that as well. And we'll even document what blade we used, the thickness of the blade. And so then someone in the prep lab can go back, and if you want, you can put the mold or cast of the chunk that was taken out back into the real fossil. You even know how thick to make your epoxy on either side if you know the blade size. And you can put it back together and it looks pretty much like it did right before you ever cut it. Sometimes we don't do that. It depends on preparator preference. Sometimes they want to leave it fully apart instead of gluing a mold back into like a real fossil. So sometimes you'll end up with a cast from before it was ever cut, a mold and cast of the piece that was taken out along with the original material. And then the slides and any of the remaining like puck that was taken out will all go back to the host museum so that other researchers can see it. So there is some loss to the blade, but it's not very much. And the molds and casts preserve the original size, and any of the structures or external features that were there before it was cut are going to be preserved with photos and 3D models and the mold and cast. So I think we learn a lot more than we lose from it. So definitely worth it.
Yeah, no, thanks for that. That was very, very detailed. Next question is, can the lines of arrested growth be affected by illness? And if so, how?
Physiologically, yes. Lines of arrested growth can be, the interpretation of those lines can be impacted by illness. So some examples in human histology, in teeth, for example, there are actual growth lines in teeth. They look a little different than the growth lines in bone because the tissue looks a little different, but you can get stress lines in teeth in humans during years of like for famines and times of hardship. And yes, presumably you can also get that in non-human animals as well. Though I don't have any slides that show anything that I would immediately interpret as pathologic. It is possible to have some lines show up that are reflective of some kind of pathology or of some like trials and tribulations, like a hard year for sure. So that's why one of the reasons why I like to have an entire section of a bone, so that I'm not just getting one small area. I want to see the whole record from the very inside margin of the bone all the way to the outside. And that way we can sort of say if anything looks unusual, that might be some indicator of pathology, and then the rest of the record is there to compare it to.
Okay, no, sounds good. The next question is kind of two that they're related, so I'll kind of put them together here. One is about the vascularity within the bone. Is that inherent when the bone is created, or does that penetrate into the bone after the fact? And then the second related question about that is, there's, in one of the profiles you showed, the kind of the corroded surface of the bone internally. Is that kind of some sort of resorption? What's happening there? And is that related to disease or stress?
Good questions. So vascularity is something that grows with the bone. So as the bone tissue is being laid down, the blood vessels are in there while the bone cells are being laid down. So that vascularity is going to be present as the bone grows, and it's going to change as the bone grows. And then any later remodeling could change older layers of bone as well. So you could get new vascular canals that come through and kind of disrupt older bone. You can get remodeling where bone cells are actually like destroyed and rebuilt, and new bone is laid down, and you end up with these donut shapes that have lots of little concentric circles around them where they are being remodeled over time. You can get remodeling for a couple different reasons. Sometimes it is muscle attachment, but that's going to be visible sort of towards the outer margin of a bone, and it'll be in a predictable place. So that's one reason that we take slides for ontogeny, ontogenetic identification. We use the middle of the bone because there's not very many muscle attachments right in the middle of like a long shafted bone. So you wouldn't really want to cut towards like the top of the femur or the bottom of the femur or the tibia, either one, because that's where muscles are attaching. And you're going to get remodeling from both muscle attachment and from stresses for sure. If there's any breaks or if there's just a lot of stress on that part of the bone, you could get a lot of remodeling, and that might kind of overprint your interpretation of the osteology and ontogeny. So that's why we go for the midshaft. There are a lot fewer of those stresses working at the midshaft. There are a lot fewer muscle connections that would come anywhere near the midshaft. So you get a much cleaner idea of what the actual just bone histology without all the other stuff overprinting on it is. Yeah, but it is something we have to account for. And there have been studies looking at like the same bone at different points along the bone and showing remodeling scars from muscle attachments or from other stresses and things like that.
Great. The next question is about, do we know if there's any change or differences in the rate of growth between different bones of the same animal?
Yes, one of my colleagues, Tom Cullen, has done a study on that fairly recently, where he compared lots of different animals and actually looked at tibia and a rib fragment and like locations on different bones of the same animal. And you can get different lag counts depending on which bone you're looking at. And there are different remodeling markers on different kinds of bones. Some people even do histology of the jaw bones of different dinosaurs. One reason I use long bones, the tibia and femur, is because in the studies that compare different bones, the femur and the tibia have a very consistent, very reliable signature at the midshaft. It's the least likely to have overprinting from anything else the animal is doing. The jaw can have a lot of overprinting because there's a lot of mechanical stress on the jaw. So I tend to veer away from using jaws. And in the same animal, you can get different lag counts. But if we're all using comparable bones, we can kind of actually compare our research to each other. So that's an important part, is not just how many legs are there, but we want to compare, okay, how mature is this animal compared to another animal? And if we use the same bone, so if we use the tibia from animal A and the tibia from animal B, we are going to be able to compare those results and say, well, this animal is more mature and this animal is less mature. It gets harder if you're trying to compare different bones of different animals to each other. So that's why we try not to do that.
Okay. Yeah, great question and good answer. We have two more questions if you have time for that.
Sure.
The first is actually a comment and a question from one of your colleagues, from Greg Funston. So he says, first off, fantastic talk. Really exciting to see interesting and unexpected pieces of the puzzle, and that the conundrum of chirostenodes is really important, because the Tyrrell specimen is often taken as the best example for that. Do you have any plans, or are there any plans to section the holotype of chirostenodes to see whether it's an adult, and alternately, what would be the other options if you cannot section it?
This is a great question. Of course, I would expect nothing less, Greg. Yeah, I would love to section the holotype. It is only hands, so I'm not sure how comparable it would be. I've not done any thin sections of like the manual phalanges of anything other than one of the ROM specimens, which we were doing for that intraskeletal study, looking at how different the interpretation was from one bone to the next in the same animal. But it would be really fantastic to get some kind of sample, and at least get an idea of relative maturity. So even if we couldn't compare it directly to the tibia results from another animal, you can still get an idea of relative maturity or immaturity, like we were talking about in some of those slides with the degree of remodeling and the vascularization changes and whatnot. So yes, that would be fantastic. I don't know if it's possible yet or not, but I would love to get a sample of that specimen. Yeah, that's one of the most exciting projects to me, I think, because we have clear evidence that some of the chirostenosis material is adult and some of it is very much not. So yeah, I'd love to be able to dive more into the material there and try to sort that out.
Okay, one final question in that, have you considered doing a morphospace or morphometric analysis for these Oviraptorosaurs, and help with that, if that would help with the taxonomy and the phylogeny for the group?
It could. I'd love to do some morphometrics with these guys. The problem so far is we don't have a ton of material to work with for a geometric morphometric analysis. And the elements we do have are very similar. So these species are not drastically different from one another. And we don't have a lot of complete material. So a morphometric analysis would be better if we had like a whole skull. But we do have plenty of dentaries, so that could be a good thing to look at to try to create some morphospace categories. The problem is the fragmentary nature. It's hard to figure out which dentaries go with which bodies because they're not preserved together. So we have hands, feet, jaws, but very few of them are preserved in association, which makes it really difficult to do any sort of analysis of how the morphology is different from one species to the next, because it's hard to know what the entire body looks like. So I'm hoping that as we get more specimens and as more discoveries are found, that we'll be able to start piecing that together and figure out whose jaws go with which feet, and get a better idea of the morpho characteristics. So what are the differences between these different species and how they look, and what can that tell us? Would be great. But we'll have to wait for some more complete specimens so we can actually pair things together a little more with more certainty. Yeah.
Thank you very much for answering all those questions. That's really good questions from our audience today. So thank you for everyone who submitted questions. I want to thank Jade again for a really cool talk about a fascinating subject. And before we conclude, again, just another shout out to next week's talk. So April 21st, Dr. Hanneke Mayor from University of Bern in Norway. Birds above the Hobbit's head, tales of a small island far, far away. So sounds like a great talk. Make sure that you come and take part in that one.